Okay, hi, I'm Rico.
Hi, I'm Kamina.
Hi, I'm Leslie,
Hi, I'm Chetna.
and we looked at the paper entitled Induction of Pluripotent Stem Cells from Mouse Embryonic and Adult Fibroblast Cultures by Defined Factors. The background and significance of this paper centers around embryonic stem cells. Embryonic stem cells come from the inner cell masses of mammalian blastocytes and are able to continue to grow and grow while maintaining pluripotency and the ability to differentiate into all three germ layers. These human embryonic stem cells could be used to treat a myriad of conditions like Parkinson's, spinal cord injuries, or diabetes. However, there are ethical concerns over the use of human embryos and issues related to tissue rejection after transplantation. So, one of the ways to help remove these concerns would be to use the patient's own cells that are generated to be pluripotent. The researchers looked back at previous research studies that showed that somatic stem cells could be induced to be pluripotent by either transferring their nuclear contents into oocytes or by being fused with embryonic stem cells. Both of these situations indicate that unfertilized eggs and embryonic stem cells have factors necessary to confer this pluripotency to somatic stem cells. Not somatic stem cells, just somatic cells. And so, knowing this, the researchers hypothesized that various transcription factors, which are involved in the maintenance of pluripotency in embryos, and genes upregulated in tumors that are responsible for the maintenance of embryo stem cell phenotypes, may allow for this induction of pluripotency within somatic cells. Examples of these would be the transcription factors OCT3/4, SOX2, or NANOG, and then genes within tumors. Examples would be STAT3, ERAS, c-MYC, KFL4, and B beta-catenin. So this led the researchers to look at the big research question of could these factors be used to reprogram somatic cells and induce pluripotent cells to help bring about that embryonic stem cell state.
Okay, so the first thing they needed to do was develop an assay for detecting pluripotency, and you can see this in Figure A. So the FBX15 locus, which is required for maintenance of pluripotency, is attached to a beta geocassette, which contains antibiotic-resistant genes. And the general idea behind this is that factors that induce pluripotency will lead to antibiotic resistance of the antibiotic G418, which was used for selection. Cells were infected using retroviruses for 24 candidate genes, and the results of this can be seen in Figure B. The left dish is just a control, and the right dish is showing cells infected with all 24 genes. So there are colonies present on this dish, and based on the system I just described to you, they are pluripotent. Further experiments in which they infected cells with only one gene actually yielded no colonies, which suggests that multiple genes are required for pluripotency. In Figure C, this is just comparing embryonic stem cells to the induced pluripotent stem cells they created based on their morphologies. Both are round in shape and have large nucleoli. And the last panel is just showing the cells they used, which were mouse embryonic fibroblasts. And this is what they look like prior to infection. Next slide, please. So next, they did a series of experiments where they infected cells with all 24 factors but one for each factor, and the results of this can be seen in Figure A. So for 10 factors, what they found is that cells either did not grow, did not have any initial growth before 10 days or no growth at all, suggesting these are important for pluripotency. So from these 10 factors, they repeated this procedure of infecting cells with all 10 factors but one, and this can be seen in Figure B. What's really interesting here is that with only 10 factors, there was a larger amount of colonies than with all 24 factors. So they narrowed this number down further from 10 factors to 4 factors that had no growth, which suggests they are really important for pluripotency. And with these 4 factors, they did further experiments as seen in Figure C, where they tried various combinations, and the only one that really yielded anything comparable to the 10 factors was all 4 factors. Next slide.
Okay, so next, RT-PCR was performed to examine whether ES cell marker genes were expressed in iPS cells. So first of all, from the figure, we can see that all the genes are activated in ES cells. And we can also see that iPS MEF4 and 10 cells expressed most of the marker genes other than ECAT1. But in iPS MEF3 clones, we can see that ECAT1, ESG1, and SOX2 weren't activated, and some genes were induced but very weak. This shows that iPS MEF4 and 10 cells are similar but not identical to ES cells. And it also shows that iPS MEF3 cells are much more different from iPS4, 10, and ES cells. Next slide. So then global gene expression profiles of ES, iPS, and other control cells were compared using DNA microarray. In the figure, the red shows upregulation and green shows downregulation of genes. So group 1 shows upregulated genes in ES and iPS. And then from group 2 and 3, we can see that iPS 4 and 10 cells are the most similar to ES cells. So this confirms that iPS cells are similar but not identical to ES cells. Next slide. Thank you. Okay, so next, in vitro embryoid bodies were formed and immunostained. So when embryoid bodies of iPS 3, 4, and 10 cells were grown in tissue culture dishes, iPS MEF4 and 10 cells attached to the dish and initiated differentiation, which can be seen in the red box. But in iPS 3, but the iPS 3 cells did not. So then after 3 days, the embryoid bodies were immunostained with alpha smooth muscle actin, alpha fetoprotein, and beta tubulin, which are markers for mesoderm, endoderm, and ectoderm respectively. And as shown in figure D, while we can see detection for all the markers in iPS 4 and 10 cells, nothing was seen for iPS MEF4 3 cells. So this confirms the pluripotency of iPS MEF4 and 10 cells. Next slide.
So now we come to conclusion. So now we can conclude that Yamanaka and his group were successful to generate induced pluripotent cells by introducing 4 transcription factors into the mouse fibroblasts using retrovirus. Also, these induced pluripotent stem cells can differentiate in vitro and in vivo even with the presence of retroviral vectors. As we know by now that among these 4 transcription factors of 3x4 and SOX2 are essential for the generation of induced pluripotent cells. And surprisingly, NANOG is dispensable. Next slide, please. Uh, so then biochemical and genetic analysis of induced pluripotent stem cells in this study found that KLF might contribute to the activation of NANO through p53 repression, and it might function as an inhibitor of NYC-induced apoptosis through the repression of p53 in the system. Then, according to the global gene expression patterns and DNA microarray analysis, iPS MEF4 cells and iPS MEF3 cells have the same origin. Also, iPS MEF4 and iPS MEF10 clones exhibit pluripotency. Also, this data confirmed iPS cells were similar but not identical to embryonic stem cells. Then this study also has a surprising finding, that is the efficient activation of FGF4 and FBX15 by the combination of the factors devoid of SOX2. Next slide, please. Then after this study, there are some unsolved questions or the questions which needs to be studied more in detail. Like first one is that although authors found that transgene reporters with other embryonic stem cell marker gene can replace with FBX15, but they still don't know whether the 4 factors can generate pluripotent cells from human somatic cells. But later in 2007, same group has reported the generation of human-induced pluripotent cells. Then there is a need of detailed analysis to enhance the understanding of transcriptional regulation in pluripotent stem cells. Then next slide, please. Then comes the significance of this study, and first one is that this finding is important, plays an important role and gives clear understanding, which will be very useful in the future to elucidate molecular nature of pluripotency and reprogramming. Then this result of this study demonstrated that the differentiation process was reversible when treated with defined factors. Also, the development of this technology created new avenues in basic research, disease modeling, and regenerative medicine. Thank you.